Metaphase. The complete disintegration of the nuclear environment marks the start of the second phase of mitosis. As the chromosome is spaced through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This then is that stage as with the morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids which are held together by the centromere. Small disc-shaped structure at the surface of the centromeres are called kinetochores. These structures are the charge attachment of spindle fiber formed by the spindle fiber to the chromosome that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at equator with one chromosome of each chromosome connected by kinetochore to spindle fiber from one pole to and its sister chromatid connected by its kinetochore to spindle fiber from opposite pole. The plane of alignment of the chromosome at metaphase is referred to as metaphase plate. The key features of the metaphase are spindle fiber attached to kinetochores of chromosome. Chromosomes are moved to spindle equator and get aligned along metaphase plate throughout the spindle fiber into both poles. Anapause at the onset of anapause, each chromosome arranged at the metaphase plate is split simultaneously under two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration toward the two opposition poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and has at the leading edge with the arms of the chromosome trailing behind. The draws in a phase stage is characterized by following key events: centromeres split and the chromatids separate. Chromatids move to opposite poles at the beginning of the final stage of karyokinesis. The chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosome can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which shows following key events: chromosome cluster opposite spindle poles and the identity is lost. The discrete element nuclear envelope developed around the chromosome cluster at each pole, forming two daughter nuclei, nucleolus, Golgi complex, and ER. From cytokinesis, mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclear karyokinesis, build the cell itself to divide into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal, this is achieved by appearance of puro in the plasma membrane. The puro gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by relatively inextensible cell, however, while therefore death undergoes cytokinesis by a different mechanism. In plants, cell formation start in the center of the cell and grow outwards to meet the existing lateral walls. The formation of new cell wall begins with the formation of the simple precursor called the cell plate that represent the middle lamella between the walls of two adjacent cells. At a time of cytoplasmic division, organelles like mitochondria and plasmids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis. As a result of a voluntary nuclear condition arises, leading to the formation of syncytium liquid in the sperm in coconut. Mitosis or the equational division is usually restricted to the diploid cells. However, in some lower plants and in some special insects, haploid cells also divide by mitosis. It is very essential to understand that the significance of this division in the life of an organism. Mitosis is usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It is therefore become essential for the cell to divide to restore the nucleus-cytoplasmic ratio. A very significant contribution of mitosis is cell repair. For the cell of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Mitosis division is the metabotic tissue. The apical and the lateral camera result in continuous growth of plant throughout the life. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a hapless set of chromosomes. Gametes are formed from specialized diploids. This is specialized kind of cell division that reduces the chromosome number of half results in production of haploid daughter cells. This kind of division is called meiosis. Meiosis is show the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across Meiosis during gametogenesis in plants and animals. This leads to formation of haplogamma. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatid at the S phase. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromosomes of homologous chromosomes. Four haploid cells are formed at the end of meiosis. Two meiotic events can be grouped under the following phases. Prophase 1. Prophase 1 of the first meiotic division typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following five phases based on the chromosome behavior. Lepton, zygote, packaging, diplotin, and glycan. During the lepton stage, the chromosome becomes gradually visible under the light microscope. The compaction of chromosome continues toward leptogenesis, followed by the second stage of meiosis 1 called the zygote. During this stage, chromosomes start pairing together, and this process association is called such pair chromosomes are called homologous chromosomes. Electron micrographs of this image stage indicate that the chromosome synapse is accompanied by the formation of the complex structure called synaptonormal complex. The complex formed by a pair of synapse homologous chromosomes is called a bivalent or a tetra. However, these are more clearly visible at the next stage. The first two stages of phase 1 are relatively short-lived compared to the next stage, that is the kiting. During this stage, four chromatids of each bivalent chromosome become distinct and clearly appears a tetra. This stage is characterized by the appearance of recombination nodules, the site at which crossover occurs between non-sister chromatids of the homologous chromosome. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also enzyme involved is called recombination. Crossing over is the combination of genetic material dikinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the mitotic spindle is assembled to prepare the homologous chromosome for separation. By end of dikinesis, the nucleus disappears and the nuclear envelope also breaks down. Dikinase preparation the transition to metaphase. Metaphase 1. The bivalent chromosome aligns the equatorial plate, the microtubule from the opposite poles of the species to kinetochores, the homologous chromosome. And the homologous chromosomes supported by sister chromatids remain associated at their centromere. Telophase 1. The nuclear membrane and the nucleolus reappear. Kinesis follows as this stage is called as tide of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus, the stage between the two sister two mitotic division interkinase and the generally short time. There is no replication DNA during interkinase. Interkinase followed by prophase 2, a much simpler prophase than prophase 1. Meiosis 2. Prophase 2. Meiosis 2 is initiated in the three cytokines usually before the chromosome have fully elongated. In contrast to meiosis, meiosis 2 resembles normal mitosis. The nuclear membrane disappears by the end of the prophase 2. The chromosome again become compact. Metaphase 2. At this stage, the chromosome align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatid. And phase 2 begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatid together, allowing them to move toward opposite poles of the cell by shortening of microtubule attached to kinetochores. Telophase 2. Meiosis ends with telophase 2, in which two groups of chromosomes occur again get enclosed by a nuclear envelope. Cytokinesis following results in the formation of tetrad of cells. Four Lloyd daughter says meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organisms. Even though the process per se paradoxically results in a reduction of chromosome number by half, it also increases genetic variability in the population of organisms from generation to the next. Variation are very important for the process of evolution. Endoplasmic reticulum. Electron microscope study of eukaryotic cell reveal the presence of the network reticulum, tiny tubular structure structured in the center that as endoplasmic reticulum has ER divides the intracellular space into two distinct compartments, luminal inside ER and extraluminal cytoplasm compartment. The ER often shows ribosome attached to the outer surface. The endoplasmic reticulum bearing a ribosome on their surface is called rough endoplasmic reticulum. In absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous, while the outer membrane of the nucleus, the smooth endoplasmic measures site for synthesis of lipid in animal cells. Lipid like steroid hormones synthesized in SER. Ribosomes. Ribosomes are the granular structure first observed under the electron microscope at this particle by George Fallon in 1953. They are composed of ribonucleic acid and protein and are not surrounded by any membrane. The eukaryotic ribosomes are 80s, while the prokaryotic ribosome are 70. Each ribosome has two subunits, larger and smaller subunit. The two subunits of 80 ribosomes 60 40s, while that of 78 ribosomes 50s 30s, and as Swebber's unit stands for sedimentation coefficient. It is indirectly measure of density, and both 70s and 80s ribosomes are composed of two subunits. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus changed by the basic dyes were given the name chromatin by Fleming. The interface nucleus of the cell, when it is not dividing, has highly extended and elaborate nucleoprotein and once or more spherical called nucleoli. Singular nucleolus electron microscopy has revealed that this nuclear envelope, which consists of the two parallel membrane with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane is usually remain continuous with the endoplasmic reticulum and also bear ribosome in it. As at a number of places, the nuclear envelope is interrupted by minute pores which are formed by fusion of two membranes. These nuclear pores are the passes through which movement of RNA and protein molecule takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Some mature cells even lack nucleus. Erythrocytes of many mammals and sheep tubes cells of vascular plants. The nuclear matrix or the nucleoplasm contains cells and chromatin. The nucleoli are spherical structure present in the nucleoplasm. The content of the nucleus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Are larger and more numeral nucleoli are present in cells actively carrying out protein synthesis. The interface has a loose and instinct network of nucleoprotein fibers called chromatin, but during different stages of cell division, cells show structural chromosome in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2-meter long head of DNA, 23 pairs chromosome. Every chromosome visible only in dividing cells essentially has a primary construction or the centromere, on the sides of which these structure chromosomes are present. Centromere holds two chromatids of the chromosome. Based on the position of the centromere, the chromosome can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. Some metacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to it and forming one extremely short and one very long arm, whereas the retrocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary construction at a constant location. This gives the appearance of the small fragment called the satellite. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 19th. Meanwhile, the chemical studies on the cellular membrane, especially in the human red blood cell, enabled the scientists to deduce the possible structure of plasma membrane. This study showed that the cell membrane is mainly composed of the lipids and protein. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and hydrophilic head towards the inner part. This ensures that a non-polar tail of saturated hydrocarbons protected from the aqueous environment. In addition to phospholipids, the membrane also contains cholesterol. Later, biochemical investigations clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipids varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral protein lie on the surface of the membrane, while integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nichols in 1970, widely accepted a fluid mosaic model. According to this, the quasi-fluid nature of the liquid enables lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intracellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of plasma membrane is the transport of the molecule across it. The membrane is structurally permeable to some molecule present on either side. The many molecule can move briefly across the membrane without any requirements of energy, and this is called the passive transfer. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient from higher concentration to the lower. Water may also move across this membrane from to lower concentration. The movement of water by diffusion is also called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate transport across the membrane. A few ion or molecule are transported across the membrane against the concentration gradient from lower to higher concentration. Such a transport is an active dependent process in which ATP is utilized, called active transport. Example: Na plus K plus form.